When you just look around, from the smallest little insect to, I don't know, a huge elephant, it's kind of mind-boggling how they all manage to create more of themselves.
Right. What's that fundamental process, that thread that links them all?
Exactly. And that fundamental secret, that's really what we're diving deep into today. Reproduction in animals. Our mission really is to unpack all this complexity, pull out the really fascinating stuff, sometimes surprising details about how life keeps going using the great material we've looked at.
Okay.
So let's maybe kick things off with something the sources really highlight. Processes like digestion, breathing, they're vital for, the individual animal, right? But reproduction, that seems to be playing a different kind of game entirely. What's its unique importance?
That's a great way to put it. Digestion keeps you alive today. Reproduction keeps the species alive tomorrow and the day after that. Imagine if organisms just stopped reproducing. Poof, they'd vanish. It ensures that similar kinds of individuals keep appearing, generation after generation. It's all about continuation.
Okay, continuation of the species. And our source material clearly lays out two fundamental ways animals achieve this. What are these two main modes?
Right. Animals reproduce either through sexual reproduction or asexual reproduction.
Yeah.
We get to definitely explore the distinct features of each one.
Okay. And this is where it gets really interesting, I think. Let's start with sexual reproduction. It's probably the one most people are, well, more familiar with.
Sure. So sexual reproduction, it begins with the fusion of male and female gametes. These are specialized reproductive cells. They join together to form a single cell, which we call a zygote.
A zygote, okay.
And it's that zygote that then develops into a whole new individual.
And our sources use humans as a really key example here, delving into the reproductive organs. So maybe starting on the male side, what are the key parts involved?
Okay, for the male reproductive system, the main components described are a pair of testes. The singular is testes, two sperm ducts, and a penis. Now, the testes are crucial because they produce the male gametes. These are called sperms, and they produce millions of them.
Millions.
Millions. And each sperm itself, it's structured very specifically. It has a head, a middle piece, and a tail. And importantly, each sperm is a single cell, containing all the usual cell components, just specialized for its job.
A single cell designed for that one purpose. And you mentioned the structure head, middle piece, tail. What's packed inside that makes it work?
Right. So the head mainly contains the nucleus. That's where the father's genetic information is. The middle piece is packed with mitochondria, which are like tiny power generators providing energy.
Energy for the tail.
Exactly. Energy for the tail to propel the sperm on its journey. It's incredibly streamlined.
Okay. And then on the female side, what are the key organs there?
So the female reproductive organs include a pair of ovaries, the oviducts, which you might know as fallopian tubes, and the uterus. The ovary is where the female gametes, called ova or eggs, are produced. In humans, typically one single matured egg is released each month into an oviduct.
Just one per month. Quite different from the millions of sperm.
Very different, yes. And the uterus, that's the organ where the baby develops. And just like the sperm, the egg is also a single cell. And what's really fascinating here, thinking about eggs, is the size variation across the animal kingdom. A human egg is tiny, really, but a hen's egg, much larger, obviously. And the source material points out the ostrich egg as being the largest single cell known.
The largest single cell, that's incredible. Okay, so we have these specialized gametes, the sperm and the egg. What's that very first crucial step where they come together?
That step is fertilization. It's the fusion of a sperm and an ovum, or egg. So when sperms reach an egg, usually only one sperm manages to fuse with it. And during that fusion, the nuclei, the cores containing the genetic material from both the sperm and the egg, merge. They form a single nucleus, and this creates the fertilized egg, which is, as we said, the zygote.
Right, the zygote, that single cell start. So what does this fusion, this merging of nuclei, actually mean for the characteristics of the new individual that develops?
Well, it's everything, really. Fertilization is literally the meeting point of the mother's contribution, the egg cell, and the father's contribution, the sperm cell. This means the new individual inherits some characteristics from the mother and some from the father. That's why you often see family resemblances.
Ah, okay. So that explains why you might have your dad's nose or your mom's eyes or something like that. It's all baked in right from that first fused cell.
Precisely. It's the combination of those two sets of genetic instructions.
Now, our source material pointed out something interesting. This fusion, this fertilization, doesn't always happen in the same place. There are two main locations.
That's right. We have internal fertilization and external fertilization. Internal fertilization is when the fusion happens inside the female body. This is what we see in humans, cows, dogs, hens, many land animals, basically.
Okay, inside the female. Makes sense.
Then there's external fertilization.
Right.
This happens outside the female body. It typically occurs in water. Think of animals like fish, starfish, and a key example mentioned is frogs.
Frogs. Okay, so they release eggs and sperm into the water.
Yes, and this brings up an interesting point mentioned in the sources. Why do animals like fish and frogs lay hundreds of eggs, while a hen, which also reproduces sexually, usually lays just one at a time?
Yeah, that's a huge difference.
Well, for frogs, for example, the eggs aren't protected by a hard shell like a hen's egg. They're quite delicate, often held together in a jelly-like substance. The female lays these eggs in the water, and then the male releases sperm over them. The sperm then have to swim through the water to reach and fertilize the eggs.
Okay, so why the massive numbers then? If they're just releasing them into the water, seems risky.
It is risky. That's exactly why the large numbers are necessary. These eggs and sperm are exposed to everything: water currents, wind, rain, changes in temperature. Plus, lots of other animals in the pond or river might just eat them. They're an easy food source.
Ah, so it's a numbers game.
Absolutely. By producing hundreds or even thousands of eggs and vast quantities of sperm, they increase the statistical probability that at least some will survive, get fertilized, and develop into new individuals. It's a strategy for overcoming those high environmental risks.
That makes sense, a sort of evolutionary bet hedging. Okay, so once fertilization has happened, whether it was internal or external, we have the zygote. What's the next stage in its journey?
Right. The zygote starts dividing. And dividing again, it basically develops into what we call an embryo. It divides repeatedly to form a ball of cells first. Then these cells begin to differentiate, organizing into groups that will eventually develop into all the different tissues and organs of the body.
So it starts specializing quite early on.
Yes. And in internal fertilization, like in humans, this developing embryo typically embeds itself into the wall of the uterus. That's where it continues its development.
And it stays there, growing.
Exactly. It gradually develops identifiable body parts. You start to see things like hands, legs, the head, eyes, ears forming.
And is there a point where we stop calling it an embryo and start calling it something else?
Yes, there is. The stage of the embryo where all the main body parts can be identified is called a fetus.
Fetus, got it.
And then when development of the fetus is complete, the mother gives birth.
Okay. But then we have animals like hens. You mentioned they have internal fertilization.
Right.
But they obviously don't give birth to live chicks. They lay eggs.
Yeah.
How does that process work?
That's a great example of the variation. So in hens, fertilization happens internally, yes. But soon after that, the zygote starts dividing as it travels down the oviduct. And as it travels, protective layers are formed around it. This includes the hard shell that we're all familiar with.
Ah, the shell forms after fertilization as it's moving.
Correct. The hen then lays this shelled egg. The embryo inside continues to develop, and it takes about 3 weeks for it to become a chick. The hen often sits on the eggs during this time, providing the necessary warmth for incubation.
Incubation, right.
Until eventually, the fully developed chick hatches or bursts out of the egg.
Fascinating. So based on this difference, giving birth versus laying eggs, our sources give us two categories for animals, right?
Exactly. Animals that give birth to live young ones, like
Yeah.
humans, cows, dogs, are called viviparous.
Viviparous, okay.
And animals that lay eggs, like hens, frogs, lizards, butterflies, those are called oviparous.
Oviparous, laying eggs, got it. Viviparous, live birth, oviparous eggs.
That's the basic distinction, yes.
So once these young ones are either born or hatched, is it always just a case of them getting bigger, like a miniature version growing up, or are there more dramatic changes involved sometimes?
It varies again. In many animals, the young ones do look like smaller versions of the adults, and they just grow larger. Think of a kitten growing into a cat. But in some animals, the young ones look completely different from the adults. The source uses the life cycle of the frog as a classic example.
Ah, the tadpole.
Exactly. It starts as an egg, hatches into a tadpole, which is the larva, looks more like a fish, breathes with gills.
Totally different.
Totally different. And then it undergoes this incredible transformation, losing the tail, developing lungs and legs to become an adult frog.
So that's not just growth, that's a complete overhaul almost. What's the term for that kind of drastic change?
That transformation from a larva into an adult through these really drastic changes is called metamorphosis.
Metamorphosis. Right, like butterflies too, I suppose.
Precisely. Egg, larva, caterpillar, pupa, adult butterfly. That's another great example of metamorphosis. But in humans, for instance, our basic body parts are there from birth, just smaller, so we don't undergo metamorphosis. Our development is more about growth and maturation.
Okay, that distinction is clear. Now, we should definitely explore the other main strategy the sources mentioned, asexual reproduction. This one involves only a single parent. How does that work in animals? Seems simpler, maybe?
It can be simpler in some ways, yes.
Oh.
One common method mentioned is budding, which you see in animals like Hydra.
Hydra, those little freshwater polyps.
That's them. In Hydra, a new individual literally develops as an outgrowth, a bud, directly from the parent's body. Eventually, this bud detaches and becomes a new independent Hydra that's genetically identical to the parent.
Wow, like a little clone just popping off?
Pretty much, yes. It's a very direct form of asexual reproduction.
Okay, and what about even simpler organisms, like, say, microscopic ones? The amoeba is mentioned.
Right, the amoeba.
Yeah.
It's a single-celled organism, and it reproduces using a method called binary fission.
Binary fission sounds computational.
Huh. Well, it just means splitting in two. The amoeba starts by dividing its nucleus, the control center with the DNA, into two identical nuclei. Then its whole body divides into two parts, and each part gets one of the nuclei.
And boom, two amoebas.
And boom, two new amoebas from the original single parent. Again, genetically identical. It's a very efficient way for single-celled organisms to multiply.
Okay, so budding and binary fission are key examples of asexual reproduction. Now, shifting gears a bit, but still on the theme of, let's say, non-standard reproduction, our source material touches on something quite modern and often talked about: test tube babies.
Hmm.
What's actually going on there? Is it really babies in test tubes?
Yeah, that term is catchy, but definitely misleading. It refers to a process called in vitro fertilization, or IVF. In vitro just means in glass, basically outside the body in a lab setting.
Ah, okay, so it's fertilization outside the body.
Exactly. It's often used in situations where, for example, a woman's oviducts might be blocked so the sperm can't naturally reach the egg inside her body.
Yeah.
In IVF, doctors collect freshly released eggs from the woman and sperm from the man. They put them together in a lab dish, not really a test tube, usually for a few hours to allow fertilization to happen.
So the zygote forms in the lab?
If fertilization is successful, yes. The resulting zygote is then allowed to develop for about a week, becoming an early embryo. Then, this embryo is carefully placed back into the mother's uterus, where, if all goes well, it implants and develops completely, just like in a natural pregnancy.
I see. So the baby develops in the mother, not a test tube. The in vitro part is just that initial fertilization step.
Precisely. The term test tube baby is really a misnomer for the whole process.
That clarifies things. And speaking of scientific intervention and really pushing the boundaries of reproduction, the sources bring up another huge story: Dolly the sheep. This wasn't just assisting reproduction. This was something else entirely, wasn't it? What was the big deal about Dolly?
Oh, Dolly was absolutely monumental. Her birth, back on July 5th, 1996, was groundbreaking because she was the first mammal to be successfully cloned from an adult somatic cell.
An adult cell, not an egg or sperm.
Correct. A somatic cell is just a regular body cell, not a reproductive cell like an egg or sperm. Before Dolly, people thought that once a cell had specialized, become a skin cell, or a muscle cell, or, in Dolly's case, a mammary gland cell, its genetic potential was kind of fixed. You couldn't get it to generate a whole new organism.
But they did.
They did. Ian Wilmot and his colleagues in Scotland proved it was possible. Dolly showed that the nucleus of an adult somatic cell could be reprogrammed to act like the nucleus of a zygote and direct the development of a complete new individual. It was a massive scientific breakthrough.
It really changed things. Can you sort of walk us through the steps they took? How did they actually make Dolly from an adult cell?
It was a really clever, though complex, procedure. First, they took a cell from the mammary gland, the udder of a female fin Dorset sheep. Let's call her the nucleus donor.
Okay, the fin Dorset.
Simultaneously, they took an unfertilized egg cell from a different sheep, a Scottish blackface ewe. Then, crucially, they removed the nucleus from this egg cell, basically leaving an empty eggshell, genetically speaking.
Right, they took out the blackface ewe's genetic material.
Exactly. Then they took the nucleus from the fin Dorset's mammary cell and inserted it into that enucleated egg cell from the Scottish blackface ewe. This reconstructed egg, now containing the fin Dorset nucleus, was stimulated to divide as if it had been fertilized, and that it was implanted into the uterus of another Scottish blackface ewe who acted as the surrogate mother.
Wow, okay. So the genetic information came entirely from the fin Dorset sheep's udder cell, but the egg cell container and the surrogate mother were Scottish blackface ewes.
That's right.
So what did Dolly look like? Who was she genetically identical to?
Dolly was genetically identical to the fin Dorset sheep, the one who donated the nucleus from the mammary gland cell. She showed absolutely no characteristics of the Scottish blackface ewe whose egg cell was used because its nucleus was removed, or the surrogate mother.
A perfect clone of the Finn Dorset.
Essentially, yes. And Dolly was, by all accounts, a healthy clone initially. She even went on to have her own offspring naturally, which was another important proof of concept. Sadly, she did develop lung disease later and died relatively young on February 14, 2003. And it's important, as the source notes, to remember that cloning is still very difficult. Many attempts fail, many cloned animals die early or are born with severe abnormalities. It's not a simple or always successful process.
A really complex story with significant ethical considerations too, I imagine. Okay, for one last fascinating glimpse into the world of reproduction, our sources mention the honeybee hive. This isn't about technology, but about social organization influencing reproduction. It sounds quite remarkable.
It really is a fantastic example of social structure shaping reproductive strategy. In a typical honeybee hive, you have thousands of bees, maybe tens of thousands. But among all those bees, there's usually only one bee that reproduces, the queen bee. She's the only one who lays eggs.
Only one female lays eggs out of thousands.
Yes, all the other female bees in the hive are worker bees. They're sterile, and their whole lives are dedicated to supporting the colony, building the honeycomb, foraging for nectar and pollen, feeding the larvae, defending the hive, and crucially, feeding and caring for the queen to keep her healthy and laying eggs.
So the entire colony is geared towards supporting that one reproductive individual.
Pretty much. It's an extreme form of reproductive specialization within a social group.
And the source mentioned something really interesting about the eggs themselves, didn't it? About fertilized versus unfertilized eggs.
Ah, yes, that's another fascinating aspect of bee reproduction. The queen lays thousands of eggs. Now, if an egg is fertilized by sperm, which the queen stores after mating, it develops into a female bee, either a future queen or a worker bee.
Okay, fertilized means female.
But if the queen lays an egg that is not fertilized, it still develops, but it hatches into a male bee, which is called a drone.
Wow, so males come from unfertilized eggs. That's wild.
It is quite unusual in the animal kingdom, known as haplodiploidy. And the worker bees play another role here too. They carefully regulate the temperature inside the hive, keeping it around 35 degrees Celsius, which is perfect for incubating all those developing eggs, whether fertilized or not.
Incredible. The whole hive functions like a reproductive superorganism almost.
That's a good way to think about it, yes.
Well, we have certainly covered a lot of ground today, a real deep dive into this amazing, diverse world of animal reproduction, from the absolute basics like the fusion of cells,
Right, those gametes coming together.
all the way through different developmental paths, eggs, live birth, metamorphosis, and even touching on cloning and IVF.
And seeing just how vital reproduction is, not just for the individual, but for the species to persist. We've seen so many different strategies, different solutions to the same fundamental challenge, and how even things that seem simple on the surface, like budding, hide some really complex biology.
Absolutely. So, thinking about everything we've discussed, the sheer range of strategies from simple fission to complex social structures like the beehive, external versus internal fertilization, cloning, what does all this diversity really tell you about life's incredible resilience and its drive to adapt and continue here on Earth? And maybe, looking forward, as our understanding of these processes, especially things like IVF and cloning, gets deeper, how might our own definitions of what's natural, what constitutes lineage or individuality, how might those ideas continue to shift and be challenged in the years to come? Something to think about.